Good afternoon, madam. Your slide is visible?
Yes, sir.
Okay.
Slide is visible.
Yes. A very good afternoon to all the respected members who are on online and also offline.
Good afternoon, sir.
Good afternoon. The next presentation which I am covering is assessment of the antiviral potential of the plant-derived bioactive compounds. I think in pharmaceutical line, in the academic line, we are not much aware about the screening of the antiviral compounds, and we have to bring into the academic and also to the R&D sector in the academic, both in the PG and also the PhD doctorate studies, in the screening of viral compounds. And we are aware of the plants bioactive compounds, how to fractionate, how to go for extraction, which parts are being used. And these things are well studied. But this presentation covers how to take up this to the viral, antiviral property of the compounds. The viral infections such as dengue or COVID-19 continues to be a major global health concern, with rapid mutation rates and emerging drug resistance limiting the effectiveness of existing antiviral therapies. Because after the scenario of the COVID-19, most of the labs have been tuned to take up the more focus uh, on the requirement to exist in the overcome the viral infections in the global health scenario. Most of the synthetic antivirals are available. However, they have it often target a single viral protein and may lead to side effects or resistance. In this context, plant-based compounds are gaining increasing attention as a promising alternative. These derived plant molecules, these plants have been used traditionally, and also in the classical text, these have been mentioned for using as an antimicrobial properties, and some other activities. These compounds, how do you take up for the using the modern technology and modern tools for the structural when the compounds having a structural complexity and ability to act on the multiple targets, that may be an immunomodulator effect and increase the immunity as immunity boosters like that. This their broad spectrum potential and generally lower toxicity makes them valuable leads in the safe, sustainable, and effective antiviral agents. Based on this concept, this one of the major target is screening of the antiviral compounds, in silico screening of the plant-derived biotic compounds against dengue and SARS-CoV, which I will be talking here. Assessment of the antiviral potential of the phytochemical compounds that comes through the in silico studies, taken up to the viral infections of the virus cell lines on COVID replicon system, and to assess the safety and toxicity of these compounds in animal model system. In the earlier presentation, I have already covered the safety and toxicity of the plant compounds, that may be acute toxicity, subacute toxicity as per the OECD guidelines. As indicated, the different guidelines to be followed. And when we take up to the antiviral compounds, what are the safety guidelines to be followed? And the laboratory should be BSL 3 facility minimum. And these guidelines to get it approved from the regulatory authority to carry out the viral studies. And this viral entry targets the viral entry at the host response and also the cellular machinery. The antiviral mechanisms, the entry inhibition, that is at the receptor blockade, prevents virus binds to host receptors. Or fission inhibition blocks membrane fission and viral entry. At the entry level itself will be inhibited, or inhibits the required pH for entry, because to want to enter into the virus want to enter into the host cell, that there is a pH regulated, that the pH inhibition, the pH change may not be possible to enter into the virus, enter into the host. The other, that is immune modulation, that is when we look into the dengue virus, that is NS1, non-structural protein NS1, prevents complement antagonism. Whereas in the SARS-CoV, that is COVID, that it restores innate immune response. that stimulates interferon pathway, that is stimulating interferon pathway boost host antiviral state. The other target is host targeted antiviral mechanisms that block host chaperones, that ER machinery, prevent proper folding of viral proteins, inhibit lipid metabolism at the membrane level, or it may modulate the autopsy or apoptosis, strive or kill infected cells. These are the several, either it may be entry inhibition or immune modulation or the host targeted antiviral mechanisms. These are the mechanisms to be targeted to work out on antiviral compounds. Why plant compounds as an antiviral? Because of structural diversity. The explanation is unique molecular scaffolds allow strong and selective interaction with the targets. Multi-target action can inhibit multiple viral proteins or host factors simultaneously. It is having low toxicity, generally safe, well tolerated due to natural origin and traditional use, and having the as immunity boosters or immunity modulators, immunostimulants, and enhance innate immunity along with the antiviral property. That is the wide availability, that is easily source of the plant material, that is a wide variety or a cultivated varieties. The plant materials are available and they're cost effective. Based on this target, when we talk about I'll be talking here the slide, that is genome of the dengue virus and the viral targets. Here the presentation is more focused on the dengue and SARS-CoV. You have to know the physiology of the virus, that is what is the genome of the virus, that is genomic RNA encoded with the capsid protein and host derived membrane and envelope protein. Once the virus wants to enter into the host cell, it is replicated into the structural proteins and non-structural proteins. These structural proteins and non-structural proteins will be focusing on the non-structural proteins, NSP. That is the non-structural protein, that is specific proteins, that is NS1, NS2A, NS2, NS3, NS4 and NS5. This NS2 is NS2A and subdivided, and NS4 is NS2A and NS4B. But we'll be focusing more on the NS1, NS2, NS3 and NS5. As shown in the earlier, clearly the slide, that is a targeted viral function, that is a nucleic acid polymerase, that is a DNA dependent or RNA dependent or RNA dependent, DNA dependent RNA polymerase. Either it has to target here, or it inhibit the protease, or integrases, or neuraminidase. These are the antiviral main targets which we will be focusing. As I said, that NS3 helicase, whether we are focusing on the NS3 helicase or NS5, NS1, that whether it is we are focusing on the envelope or a membrane or a capsid. Because the virus, when we said that the genome, that is a capsid C or entrapped in the envelope and it is covered by the membrane, and that may be the genome when it is entered into the host cells, it is replicated into the non-structural proteins, that is NS1, NS3, NS1 to NS5. But we will be focusing on NS1, NS3 and NS5. This is specific to the dengue. In the case of a dengue, the NS3 and NS5 RNA polymerase are considered the prime target for antiviral drug therapy dependent. Based on the background of the literature, we are very clear based on the physiology and based on the multiplication, based on the genome of the viral targets. We are focused on the NS3 and NS5 RNA polymerase. The 3D structures of the dengue virus NS5 and NS3 proteins are downloaded from the PDB data bank. And these structures are based on the high resolution and contains bound inhibitors. When we look into the compounds that is existing in the market, it's Monsudevir is a registered compound as an antiviral in National Center for Biotechnology Information. PubChem, the drug is under evaluation, but to date, no structural compound, no natural compound has been reported against dengue. Based on this data and based on this information, we taken up more in the compounds of the antiviral to focus more on the dengue. And here, as I said in the earlier presentation, there are, when we take about the specific plant, there are 4 specific biological markers or the chemical markers to be quantified, quantitative and qualitative analysis to be made. And which part of the plant, what is the season, what is the, in which area, in the geographical area location, what is the solvents that are being used for extraction, and what are the phytocompounds that are present in the each plant. We have listed the plant like papaya plant, which has been used in papaya. In dengue, most of the plant that papaya leaves are being used, that a papain is there that lives, and mostly in the methanol and ethanolic extract. In the Tinospora cardifolia, this is also called as Gudici cardifolicides A, that is existing in the stem. These are the plants which have been taken from the public domain along with the compounds, along with the part used, and what is the solvent that are being used. And we have taken initially, these are the compounds and listed out. There are more than 3,000 compounds has been taken along with the part and the seed and the solvent along with the marker compounds. And the docking studies has been made. In silico studies has been made using the software tools against NS3 proteins. Based on the docking scores, we see the docking score is high, in which quercetin is minus 7.851. Like that, we have studied the docking score of the different plant compounds, which these plant compounds are existing in the plants which I listed in the earlier slide. Based on these slides, we can quantify the marker compound from that plant, isolate it and extract, and quantify it and the fingerprinting profile if you want to bring into the phytopharmaceutical regulation or Ayurveda regulation, Ayush regulation, what are the regulations you want to put into the marketing point of view. Those regulations to be followed to bring the product. And when we look into the phytopharmaceutical product point of view, there are 4 specific biochemical markers of the single plant to be quantified. And these compounds has been taken for the subsequent in silico studies and taken against non-structural protein 3, NS3. And the docking score has been recorded. This is a software analysis and the different compounds we have taken. And the docking results also studied against NS5. From this, we can understand that NS3 and NS5, there is a significant activity which is having the docking score of the high value in which plant and which specific marker based on the negative scoring. That is, when we take a minus 10.861, when it is minus, it is more high significant activity. Evaluation of the anti-JNTUK compound, the total of 3,425 compounds were collected from the Indian antiviral plants using Indian medicinal plants, biochemistry, and therapeutics database. Initially, 300 compounds were subjected for in silico screening. Top 10 compounds has been docking score and the binding free energy were tested against anti-dengue activity. When we look into the thymos vulgaris, that is the compound thymol, the docking score is minus 7.2, the binding energy is minus 6.8. These are the top 10 compounds among the plants of 300 compounds which we have taken up, we have listed here. These studies under in our laboratory is ongoing studies. And these compounds have been taken based on the docking score. 4 compounds, that thymol, camphene, carbol, beta carotene, were initially selected for anti-dengue testing. Then subjected for the cytotoxicity study, the MPT assay performed to assay it on toxicity studies on cell lines. And antiviral testing models on the Vera cell lines, initial screening against dengue, flow cytometry analysis, and a plaque assay. These are the specific standard models which have been established. When we look into the toxic concentration and maximum non-toxic concentrations, when we look into the thymol CC50, that is 73.1 micromoles. Based on the MNTC 80 and thymol with 20 microliters, camphene with 3 micromoles, carbol with 120 micromoles, and carotene with 200 compounds used for further studies, which the data has been generated in the line of the toxicity concentration of the find the MNTC compound, that is maximum non-toxic concentration. The concentration dependent virucidal activity of the phytochemical compounds like dengue virus 2, DNB virus, that is thymol, camphene, carbachol, and beta carotene. The viral infectivity, the infectivity and percentage of inhibition, there is a dose response increase. When there is increase in the dose, there is viral infection inhibition and percentage has been increased. In continuation of these studies, we studied about the virucidal activity of the phytochemical compounds against dengue virus. The studies looks very simple, but it's very complex, and safety protocols to be followed. And the lab should be well established under the biosafety lab 3 to take up the studies in the infectious models. And when these preliminary studies has been taken, because this lab is under under development, and these are the preliminary studies which we have taken, because the request from you from Bhavani madam and Sridevi, they have asked us to give the second presentation. I thought, why don't we inject the antiviral studies in the pharmacy faculty point of view, which the demand of the nation, of the country, which every lab of the national laboratories are focusing on the antiviral studies. And the most of the studies that has been taken for the testing against the compounds to be explored from the Indian context, that is most of the traditional drugs and Ayurvedic plants and the medicinal plants, which as we are very much enriched. Those compounds are well known, and those compounds, why don't we subject for the antiviral studies against the most infectious diseases, which is very highly prone and need of the country and the demand, that is dengue and also the corona. In this context, we have taken the preliminary studies now. The impact of the compound and attachment at the entry of the dengue virus on Vera cell lines, that is plaque assay, and the four potential compounds we have taken: the thymol, camphene, and carbachol, and beta carotene. Whether it has to be the entry may be inhibited or attachment may be inhibited, the percentage of entry and attachment was studied based on the specific standard assays. Inhibition was calculated as the number of the foci formed in respect to the treated and untreated cells, and 20 micromoles, and the different micromoles, the data was being generated. In line of this is the anti-dengue activity of Acranthus aspera, that is aerial and root part extract. The methanolic extract of Acranthus aspera was tested against dengue infected Vera cell lines under in vitro conditions. The bioactive compounds were downloaded and docked against NS5 and NS3 using in silico docking, and the cytotoxicity test by MTTSA has been performed. The IC50 calculation by flow cytometry based on the virus reduction and neutralization assay has been performed. And based on this data, the 25 micrograms per ml of the root extract and the aerial part 25 IC50 was been more than 25 microgram per ml has been recorded. Based on this background of the data, we went for the protein targets in SARS-CoV-2. It has to either interact with at the entry inhibition at the spike protein itself, or the polyprotein cleavage at the main protein, that is main protein, that is M-Pro or 3CL-Pro, or once it is cleaved, or immune evasion, that is a papain linked protease PL-Pro. That is the viral RNA synthesis, either it has to interfere with the inhibit the RNA dependent RNA polymerase, RDRP, or it has to interfere at the assembly and replication of the complex formation at the nucleocapsid. These are the 5 steps which the targeted compounds of the natural products or the synthetic compounds, which the synthetic the drug to be identified to set to be as a SARS-CoV-2. It has to act these 5 mechanisms, which has to be either at the entry level, or at the level of the cleavage of the polyprotein, or the cleavage at the immune invasion, or the viral CNA RNA synthesis, or at the assembly level and replication. And these plant bioactive compounds again subjected against SARS M-Pro. As I said, that is M-Pro, that is you see main protease. That main protease, this 3,425 compounds has been taken for the database and subjected these compounds against docking analysis by the bioinformatics tools against the main protease, SARS-CoV main protease, that is PDP-6LU7. The docking score and the binding energy and interaction amino acids were compared with those of the available antiviral drugs, niflunomide, which is reported an anti-HIV drug, and has been used for SARS-CoV infection. The top 3 compounds were then selected. Because when we look into the docking score of nilfenware as a reference compound, that is minus 6.139, and the binding energy is minus 77.128, and the interfering amino acid, which is very clearly reported. From this, we can say that these are the top 3 compounds, which is the docking score is higher than that of the reference compound, that is colchicide, cyanidin 3-O, rutinidine, and thiacinin B. These are the binding energy is more is 97.134. Based on this compound, the M protease activity of one of the plant extract bioactive compound against SARS-CoV has been tested. The main protease is the protein-based enzyme of coronavirus that normally breaks the polyprotein into functional proteins for viral replication in the host cells. The phytochemical extraction of these compounds has been fingerprinting profile has been taken, and these were subjected for the M protease activity. And this extract that IC50 is 114.05 microgram per ml. From this, we can understand that the plant is having antiviral property, and the fingerprinting profile of the phytochemical markers has been quantified, and profile has been the compounds has been identified by the NMR, H1MR, and these activity compounds were taken for the binding energy. These are the several compounds like arbiroside A, arbiroside C, arbiroside C, and like butylenic acid, nicotophyllene, and astragalin. These are the binding energy is very high when we look into the nicotofluorine and astragalin, and the IC50 value is 0.15 and 0.033, and the binding energy versus number of the hydrogen bonds has been taken, and it has been recorded, and a detailed study has been made in this line of investigation. And when we look into the heat map showing the binding energy kilocalories per mole of the compounds with the white type and Omicron, these are the compounds. Then these binding energies has been recorded, and the binding of Omicron with the M protein of arbiroside A or butylenic acid and nicotofluorine, and the in silico study is very clearly indicated by screening more than 3,000 compounds. We have come out with only less than 10 compounds. And this is one of the slide which we have taken up for the we have got from the USA. This is that is SARS-CoV plasmid details. When we look into the SARS-CoV plasmid detail, there is a wild type virus. It is a non-infection replicon was obtained from the USA, and the schematic diagram of the reporter of the replicon genome structure, replicon, and Hg mRNA production. The spike and envelope membrane, that is whereas in the wildlife viral genome, were deleted and replaced with the luciferase. When we look into the main point here we have to focus is the spike protein was replaced with the luciferin or a green fluorescent protein. There it is non-infectious protein replicon. Respected, the full length replicon of the cDNA was flanked by a T7 promoter and a poly A and HDB ribosome and a T7 terminator cassette, and it was published. And these non-infectious mean replicon was obtained, and we no need to go for the biosafety laboratory 3. And presently we are holding the biosafety laboratory, and we are holding this SARS-CoV wild type non-infectious mini replicon, which we are testing our most of the antiviral compounds on this replicon against SARS-CoV. This is the last slide which I am presenting. The plants bioactive compounds against SARS-CoV, which the lab is under investigation, which they taken several compounds. The non-infectious SARS-CoV replicon was transformed to E. coli, and through the chloramphenicol resistant strains have been used. The positive colony taken for the plasmid isolation and restricted by some SWA1 restriction enzyme. In the gel image, LAN M, that is LADAR 1 kb, and the LAN 1 is unrestricted plasmid, LAN 2 is a restricted plasmid. The compound selected for in silico studies will be screened using the non-infectious plasmid in vitro studies. These are the, because this presentation covers in continuation of the first presentation, the compounds not only screening for the biological activity for the therapeutic purpose, The compounds also taken for this compound for the antiviral in the second presentation which we are given, specifically focusing on the antiviral. These compounds not only taking for the directly blindly we should not go. We have taken this compound for the in silico studies, followed by taking up into the in vitro studies in the cell lines. In continuation, we taken up to these studies will be taken for the animal model system. Thank you for very much for giving an opportunity, and because the participants who are here, because I'm not much aware about the participants who are joined in the stream of science only in the it is restricted to the pharmacy or not, but because my as I am a pharmacy background, I have been but were driving in the line of national laboratory. We have taken up the problems and because these things we have to know in the graduation or the post graduation, the faculty point of view, which we can blend the students in this line of area, which it will be useful for their career and also the academic growth and the requirement of the nation. Thank you, madam, for giving an opportunity here. If any questions, I can help you.
Sir, anybody, any questions are there from the audience? You can interact with sir.
Madam, how many faculty are there, madam? How many number join here?
50 faculty members from different institutions in and around colleges in the Krishna district from our site, sir.
Yes, madam.
And I am from procuring students, we have also included.
Okay.
40 plus, 48 plus, like that faculty have attended, sir, this session.
Okay, very nice, very nice.
So very elaborative and
because what else the presentation I have given, madam, because it is not because the wide scope of opportunities they has to know when we are working in the pharmacy disciplines, because as I am also the pharmacy graduate, how we can take up the issues from the not only specifically to the branch level at the departments, we can multidisciplinary approach of several disciplines. We have to take up the studies as I shown from the pharmaceutical chemistry to the pharmaceutics and even including the antimicrobial and antiviral studies. There are several studies which we have to be taken into the quorum, madam. This is the main area.
Yes, sir. Definitely, the session will help us, sir, to take lead in selecting the projects for students or going for selecting what type of studies can be done in future and strengthening our study.
Yes,
for good publications, for getting good publications. And I think we can have in future also rapport with you in particular, getting the studies, how to
sure,
carry out.
From my side, I can say that what is the help we need from the lab point of view or in scientific strengthening the R&D activities, which you individual colleges they may be facing some on their limitations. Those limitations can be overcome by collaborations.
Yes, sir. Sure, sir. Thank you, sir.
Yes.
And in today's talk, you have explained very nicely, sir, about the assessment of antiviral potential of plant-derived bioactive compounds for dengue and SARS-CoV. So you were telling 3,425 compounds were selected from Indian antiviral plants using the database IMPP80, Phytochemical and Therapeutic Database. Excellent, sir. From that, again, top 10 compounds were under the study. You said Acarantha aspera for dengue study, where the toxic concentrations and maximum non-toxic concentrations you have shown, and in silico screening from the docking scores, binding energies, and interacting with the amino acids, those scores, by taking those scores, you have gone through the in silico screening. And you have also said that anti-empro activity, that is main protease activity of Nictanthus arbotritis for SARS-CoV. Excellent, sir. A heat map and something bind energies by using Omicron you have shown. So very updated knowledge, sir. This we have procured from you. A prolonged and extensive talk for 2 sessions by you, sir. They are excellent. And we have got so much updated knowledge from you. Thank you for sparing your time and a lot of patience. You have explained all the things. Thank you very much, sir.
Thank you, madam. Thank you very much to each and everyone. Bhavani, madam, Jay, madam. Vijaya Institute of Pharmaceutical Sciences for Women, Vijayawada, and
Sir.
also the School of Pharmaceutical Sciences and Technology, JNTUK, Kakinada. I must be
Yes, sir.
grateful. I must be grateful and acknowledge each and every participant of the college faculty who has come here, and very happy to associate each and everyone, madam. Thank you for giving an opportunity here.
Thank you, sir. Thank you.
Tomorrow, the session will start sharply by 10 a.m. I request all the faculty members to be ready in the LH1 by 9:30. Thank you. Attendance is must. 80% minimum of 80% attendance is compulsory. Just please keep it in mind and please avail the sessions.
Madam, now.